Good day. My name is Ms Prudence Mtsweni. The title of my presentation is Applied Microbiology to Improve Food Security in South Africa, my research journey. So we know that food insecurity is a global challenge that needs to be met with development of better food production system, education and conflict resolution and other impactful world hunger solutions. So for us to have food security, it means that everyone will have to have enough food and nutrition, not only enough food, but also nutritious food. So South Africa export market has always been confronted with losses due to poor quality of the fruit being due to diseases. The diseases caused by Clostridium, such as pre and post-harvest diseases of variety of crops, leading to crop losses worldwide. And the diseases, the post-harvest diseases, it does not only affect commercial farmers, but it also affects the smallholder farmers who depend on the trade to support and sustain their families. So therefore, the aim of the study is to assess the presence of the and the pathogenicity of Clostridium causing anthracnose of papaya in South Africa, and also cross-inoculate the pathogenic strains to other subtropical fruits, because these fruits they always planted in close proximity, and also when they are packed in the markets, they're also packed in close proximity. So we want to determine if the strains that are able to cause the disease on papaya are also able to cause the disease on other subtropical fruits like papaya or banana, avocado, and mango, and also develop the species-specific molecular identification too. So we've sampled in 3 provinces, Limpopo, Mpumalanga, and KZN in Mozambique. And we did the molecular identification to identify the strains, and also we did the molecular identification like DNA extraction, PCR, and sequencing, and we compared our sequences with those that are in the gene bank of how they relate to them, and also we did the phylogenetic analysis to check how they relate, and also how they separate from those that are also in the gene bank. And we've tested the strains for pathogenicity to test if the strains they serve as saprophytes or they were able to cause disease on the papaya fruits, and also cross-inoculate on banana, avocado, and mango. And we designed specific primers, species-specific primers for the detection of these plant pathogens, and we've optimized the detection too. And as a result, we've found 6 Clostridium species that have not been reported in South Africa. And these species were found to be pathogenic on papaya and on other fruits. And the species-specific primers design shown a genetic variation and separated species within the species complex, which is a challenge with Clostridium, because the primers or the barcode genes that have been used for Clostridium previously have not been able to separate the species within the species complex, which has led to species being misidentified. And so we found that the gene that we've used with this, which is partial actin, is able or it can be used as an ideal marker for barcoding of Clostridium species. So these findings will assist in the management of post-harvest diseases by developing proper disease control mechanism, and the newly designed primers can be used by DLAD in regulation of the import and export market, because when the fruits or the crops are about to be exported, they can be tested because after they've been harvested, and to test if they are infected with this Coritrichum, which causes post-harvest, and then make management of or disease management strategies can be applied. And also because some other species in other countries, they are regarded as species of quarantine importance, and therefore it can be used as